When I was a young boy, I used to gaze through the microscope of my father at the insects in amber that he kept in the house. And they were remarkably well preserved, morphologically just phenomenal. And we used to imagine that someday they would actually come to life and they would crawl out of the resin, and if they could, they would fly away. If you had asked me 10 years ago whether or not we'd ever be able to actually sequence the genome of extinct animals, I would have told you, it's unlikely. If you had asked whether or not we would actually be able to revive an extinct species, I would have said, pipe dream. But I'm actually standing here today, amazingly, to tell you that not only are the sequencing of extinct genomes a possibility, are actually a modern-day reality, but the revival of an extinct species are actually within reach. Maybe not from the insects in amber. In fact, this mosquito was actually used for the inspiration for Jurassic Park, but from woolly mammoths, the well-preserved remains of woolly mammoths in the permafrost. Woolies are a particularly interesting quintessential image of the Ice Age. They were large, they were hairy, they had large tusks, and we seem to have a very deep connection with them like we do with elephants. Maybe it's because elephants share many things in common with us. They bury their dead, they educate the next of kin, they have social knits that are very close, or maybe it's actually because we're bound by deep time, because elephants, like us, share their origins in Africa some 7 million years ago. And as habitats changed and environments changed, we actually, like the elephants, migrated out into Europe and Asia. So the first large mammoth that appears on the scene is Myriadonalis, which was standing 4 meters tall, weighing about 10 tons, and was a woodland-adapted species and spread from Western Europe clear across Central Asia, across the Bering Land Bridge, and into parts of North America. And then again, as climate changed, as it always does, and new habitats opened up, we had the arrival of a steppe-adapted species called Troglotherids in Central Asia, pushing Myriadonalis out into Western Europe. And the open grassland savannas of North America opened up, leading to the Columbian mammoth, a large hairless species in North America. And it was really only about 500,000 years later that we had the arrival of the woolly, the one that we all know and love so much, spreading from an East Beringian point of origin across Central Asia, again pushing the Troglotherids out through Central Europe, and over sort of hundreds of thousands of years migrating back and forth across the Bering Land Bridge during times of glacial peaks, and coming into direct contact with the Columbian relatives living in the south. And there they survive over hundreds of thousands of years during traumatic climatic shifts. So there's a highly plastic animal dealing with great transitions in temperature and environment and doing very, very well. And there they survive on the mainland till about 10,000 years ago, and actually surprisingly on the small islands off of Siberia and Alaska till about 3,000 years ago. So Egyptians are building pyramids and woolies are still living on islands. And then they disappear. Like 99% of all the animals that have once lived, they go extinct, likely due to a warming climate and fast encroaching dense forests that are migrating north, and also, as the late great Paul Martin once put it, probably Pleistocene overkill, so the large game hunters that took them down. Fortunately, we find millions of the remains strewn across the permafrost, buried deep in Siberia and Alaska, and we can actually go up there and actually take them out. And the preservation is, again, like those insects and animal, phenomenal. So you have teeth, bones with blood, which look like blood, you have hair, and you have intact carcasses or heads which still have brains in them. So the preservation and the survival of DNA depends on many factors, and I have to admit, most of which we still don't quite understand. But depending upon when an organism dies and how quickly he's buried, the depth of that burial, the constancy of the temperature of that burial environment will ultimately dictate how long DNA will survive over geologically meaningful time frames. And it's probably surprising to many of you sitting in this room that it's not the time that matters, it's not the length of preservation, it's the consistency of the temperature of that preservation that matters most. So if we were to go deep now within the bones and the teeth that actually survived the fossilization process, the DNA, which was once intact, tightly wrapped around histone proteins, is now under attack by the bacteria that lived symbiotically with the mammoth for years during its lifetime. So those bacteria, along with the environmental bacteria, free water and oxygen, actually break apart the DNA into smaller and smaller and smaller DNA fragments until all you have are fragments that range from 10 base pairs to, in the best case scenarios, a few hundred base pairs in length. So most fossils out there in the fossil record are actually completely devoid of all organic signatures. But a few of them actually have DNA fragments that survive for thousands, even a few millions of years in time. And using state-of-the-art cleanroom technology, we've devised ways that we can actually pull these DNAs away from all the rest of the gunk in there. And it's not surprising to any of you sitting in the room that if I take a mammoth bone or a tooth and I extract its DNA, that I'll get mammoth DNA. But I'll also get all the bacteria that once lived with the mammoth. And more complicated, I'll get all the DNA that survived in that environment with it, so the bacteria, the fungi, and so on and so forth. Not surprising then again that a mammoth preserved in the permafrost will have something on the order of 50% of its DNA being mammoth, whereas something like the Colombian mammoth living in a temperate and buried in a temperate environment over its laying in will only have 3 to 10% endogenous. But we've come up with very clever ways that we can actually capture and discriminate the mammoth from the non-mammoth DNA. And with the advances in high-throughput sequencing, we can actually pull out and bioinformatically rejig all these small mammoth fragments and place them onto a backbone of an Asian or African elephant chromosome. And so by doing that, we can actually get all the little points that discriminate between a mammoth and an Asian elephant. And what do we know then about a mammoth? Well, the mammoth genome is almost at full completion, and we know that it's actually really big. It's mammoth. So a hominid genome is about 3 billion base pairs, but an elephant and mammoth genome is about 2 billion base pairs larger. And most of that is composed of small, repetitive DNAs that make it very difficult to actually rejig the entire structure of the genome. So having this information allows us to answer one of the interesting relationship questions between mammoths and their living relatives, the African and the Asian elephant, all of which shared an ancestor 7 million years ago. But the genome of the mammoth shows it to share a most recent common ancestor with Asian elephants about 6 million years ago, so slightly closer to the Asian elephant. With advances in ancient DNA technology, we can actually now start to begin to sequence the genomes of those other extinct mammoth forms that I mentioned. And I just wanted to talk about 2 of them, the woolly and the Colombian mammoth, both of which were living very close to each other during glacial peaks. So when the glaciers were massive in North America, the woolies were pushed into these subglacial ecotones and came into contact with their relatives living to the south. And there they shared refugia, and a little bit more than the refugia, it turns out. It looks like they were interbreeding. And that this is not an uncommon feature in proboscideans, because it turns out that large savanna male elephants will outcompete the smaller forest elephants for their females. So large, hairless Colombians outcompeting the smaller male woolies. It reminds me a bit of high school, unfortunately. So this is not trivial, given the idea that we want to revive extinct species, because it turns out that an African and an Asian elephant can actually interbreed and have live young. And this has actually occurred by accident in a zoo in Chester, UK in 1978. So that means we can actually take Asian elephant chromosomes, modify them at all those positions we've actually now been able to discriminate with the mammoth genome. We can put that into an enucleated cell, differentiate that into a stem cell, subsequently differentiate that maybe into a sperm, artificially inseminate an Asian elephant egg, and over a long and arduous procedure, actually bring back something that looks like this. Now, this wouldn't be an exact replica because the short DNA fragments that I told you about will prevent us from building the exact structure, but it would make something that looked and felt very much like a woolly mammoth did. Now, when I bring up this with my friends, we often talk about, well, where would you put it? Where are you going to house a mammoth? There's no climates or habitats suitable. Well, that's not actually the case. It turns out that there are swaths of habitat in the north of Siberia and Yukon that actually could house a mammoth. Remember, this was a highly plastic animal that lived over tremendous climate variation. So this landscape would be easily able to house it. And I have to admit that there isn't a part of the child in me, the boy in me, that would love to see these majestic creatures walk across the permafrost of the north once again. But I do have to admit that part of the adult in me sometimes wonder not whether we should. Thank you very much.
Don't go away. You've left us with a question. I'm sure everyone is asking this. When you say, should we, it feels like you're reticent there. And yet you've given us a vision of it being so possible. What's your reticence?
I don't think it's reticence. I think it's just that we have to think very deeply about the implications, ramifications of our actions. And so as long as we have good, deep discussion like we're having now, I think that we can come to a very good solution as to why to do it. But I just want to make sure that we spend time thinking about why we're doing it first.
Perfect. Perfect answer. Thank you very much, Hendrik.
Thank you.